Hello everyone. Today we're going to cover Objective 3 from the Day 15, Day 16 note-taking guide that covers Chapter 17. In the first two objectives, we talked about how we could take a gene and go from a gene to a messenger RNA to a protein and how we could use the genetic code. And then we went into more of the essentials, the molecular aspect of transcription. And then today, we're going to pick up from that story where we made the messenger RNA, and in Objective 3, we're going to modify that messenger RNA. And then in the next podcast, the last one for Objective 4, we're going to talk about the proteins involved in translation. But let's go ahead and tackle Objective 3 here, which is shown here in your study guide here. And we're going to talk about modifying the messenger RNA, the three different ways it's modified. We're going to talk about why that's really important and it occurs. We're going to talk about something called RNA splicing and alternative RNA splicing. So let's go ahead and get started. So I think it's a good idea here to kind of talk a little bit about where we've been. So remember, starting with the central dogma is a good place to kind of start this discussion. Remember, we went from DNA to RNA and then to protein. And we said the process of going from DNA to RNA is called transcription. And I'm going to put my abbreviation of TXN. And then from RNA to protein, we're going to talk about translation. And we're still not going to talk about that quite yet, but we'll get there with Objective 4. Now remember, DNA, we could represent that with our double-stranded DNA. We could also show RNA as this single strand of nucleic acids. And remember, we should always include our 5' and 3' ends. And we could even put that up on here on our DNA. Okay, it's always important to include our polarity so we know kind of our landmarks, like the beginning and end of a sentence. So it's really important. Our protein, remember, we talked about how we would have our amino terminus at one end. We would have our protein sequence here. And it would end with a carboxy terminus at the far right side. And we haven't talked a whole lot about it, but after this protein is made, it's going to become modified in various ways to make a fully folded functional protein. And we'll talk more about that later. But really where we want to focus on right now is where we left off with our last podcast. And we left off our last podcast with our friend the messenger RNA here. It had just been transcribed and released from the DNA and the RNA polymerase, and it's just floating there, for lack of a better way to describe it. And in prokaryotes, or in bacteria, this messenger RNA is immediately taken to this next step and translated into a protein. But in eukaryotes, this is specific to eukaryotes. So let's go and write eukaryotes up here, because what we're going to talk about is only seen in eukaryotes. That is, cells like ours, animal cells, plant cells, fungi. And before this messenger RNA can leave the nucleus and be translated into protein in the cytoplasm, three modifications have to occur. And we're going to talk about all of them, but let's go ahead and list them here. A G cap has to be put on the 5' end of it. A poly A tail has to be put at the 3' end. And then the last thing is it needs to be spliced. And that's a difficult concept, but we'll tackle that here in a little bit. Okay, let's go to a fresh page here. And let's go ahead and draw our DNA, just to give us a reference point here. And again, we always want to put our identifiers on it so we know which direction we're going. And we know the RNA polymerase enzyme is going to bind at the promoter and move in this direction. And as it's moving in that direction, a messenger RNA is going to be produced. And so from this standpoint, let's go ahead and start talking about how it's going to be modified. The very first thing that happens, in fact, before even this big line is drawn, the moment the RNA is starting to be produced from the DNA at the 5' end, so this is a 5', and remember the reason we know this is 5' is because it's going to be anti-parallel with this 3' end. So this is a 5' end, It's the first part that's formed, and we put on what's known as a modified guanine that gets attached right to this 5' end. We can just call this a G cap. That's what it's often referred to, and that's what we'll call it too, a G cap. Now this RNA polymerase is going to continue all the way down here until it hits that terminator sequence. And when it hits the terminator sequence, remember the RNA polymerase is going to fall off, as well as all those transcription factors that aren't drawn here, but they fall off too. And what's released down here is our full messenger RNA. So let's go ahead and draw that right here. Well, actually, let's put a cross X through this here. I'm going to draw it a slightly different way. Mainly I want to leave some room down here. Okay, so let's redraw this here. And remember, we've already put on our G cap at the 5' end. So let's put it back here. Now the next thing that's added to this RNA molecule that's just been produced is what's called a poly A tail. And the A stands for adenine. So let's go ahead and put that on the end here. And we're just going to put a string of A's, adenines. We can have a few here, but a typical messenger RNA is going to have 50 to hundreds of these adenines added to the 3' end. I'm just going to draw what I have here, 5, but recognize that this is usually quite a bit more. And it's still the 3' end. So what we have after the first two forms of modification, the 5' cap, the poly A tail, and the messenger RNA here. Now remember, this messenger RNA is going to have all the nucleotides on it, all the RNA nucleotides that are going to code for that protein. So we're going to have our AUG at the very first part here, and you're going to have all the other nucleotides ending with one of the 3 stop codons, like UAA is one of the 3. UAG is another one. So there's 3 of these, but we'll just put UAA here now. So now this messenger RNA is almost ready to be sent to the cytoplasm to make a protein. The only other thing that has to happen is it needs to be spliced. And when we talk about splicing, I'm going to go to another slide to go through it because it's a little bit more complicated. But before we do that, let's spend just a little bit of time talking about why we add this the 5' G cap and the poly A tail. And really, there are 3 main reasons, and each of these reasons applies to both the G cap and the poly A tail. This first one I'm going to write down is stability. Stability, protection, life of the messenger RNA is all determined by the presence of the 5' G cap and the poly A tail. The reason for that is once this messenger RNA is made, various enzymes can begin to degrade the messenger RNA. However, those enzymes are very specific. Remember how we talked about how enzymes have an active site and they recognize a very specific substrate? So for instance, if this messenger RNA did not have this 5' cap, in fact, why don't we just draw one here that's not been modified? We can draw it right below it. So here's a messenger RNA that's not modified. Well, you may have these enzymes that recognize RNA. So let's just draw an enzyme here that can recognize RNA molecules. However, it's very specific to this kind of RNA. It cannot bind to this modified G protein here. Modified G nucleotide here because of enzyme specificity. So this protects this messenger RNA from being degraded at the 5' end because this enzyme is no longer functional here. It also provides stability for the poly A tail. There are enzymes that can bind to this poly A tail and begin to degrade it. It's pretty slow because in addition to this poly A tail being here, we know various proteins will also bind to the poly A tail to give that end of that messenger RNA a lot more stability. But over time, the adenines slowly get degraded. And at some point, once all that happens, then when the poly A tail is gone, the rest of the messenger RNA is pretty much rapidly degraded. In fact, we can tell a lot about how long a messenger RNA is inside of a cytoplasm based upon how long its poly A tail is. It pretty much is the lifeline of the messenger RNA. The longer the poly A tail, the longer lived that messenger RNA will be compared to a shorter poly A tail. The other purpose of these modifications is for nuclear export. Without these modifications on the messenger RNA, it would be unable to leave the nucleus. And remember, it has to leave the nucleus in order to make a protein from it. So they aid in nuclear export. Then the last thing we should talk about real briefly, we'll come back to this later, is that it's these both the 5' GCAP and the poly A tail play an incredibly important role in translation. And they play a role in translation because they bind the ribosomes. We haven't talked about the molecular process of translation yet. We will in the very last objective, so the next podcast. But we could talk a little bit about it in the sense that once this, we'll go ahead up to our good friend we wrote up here, the fully modified one. What happens is that we know that the ribosome is going to bind here at the AUG. It recognizes this start codon because of the 5' GCAP. And then what happens is this poly A tail, it's hard to draw this, so I'm not even going to try it, but it kind of loops around and binds to the ribosome as well and helps position it for translation. So both the 5' GCAP and the poly A tail are important in translation by binding to and stabilizing the ribosome. And we'll talk much more about the ribosome and translation, as I said, in the next podcast. Now, on the next slide, let's talk about this third modification, and that is splicing. We've just talked about GCAP, poly A tail, and now let's talk about splicing. This is an incredibly important thing that happens with eukaryotic messenger RNAs. Again, this does not happen with prokaryotic messenger RNAs. So let's begin by drawing, just a random gene. And we know that our RNA polymerase will bind again to the promoter and begin making messenger RNA. We've already talked about that to a great extent. And we said we had this transcription unit here that makes the messenger RNA that will ultimately make the protein. And that's pretty much all we said. But it's really important what this looks like here. So we want to introduce a few new terms for you. Piece of DNA here, this gene, can be divided up into several subunits. And these subunits are called exons. I'm going to put an E here for an exon and then 1 because this is exon 1. And I'm going to write the word exon here. And then right next to it is going to be intron 1. I'm going to write intron here so you know what I'm talking about. And then next to it we have an exon 2. And next to that we have intron 2. And next to that we have exon 3. And we can have hundreds of introns and hundreds of exons. But for simplicity, I'm just going to stop here with 3 exons and 2 introns. And then we have our terminator sequence that we've already talked about. This is going to signal when to stop transcription. Okay, so the RNA polymerase moves along here, and it makes the messenger RNA. So let's draw this messenger RNA real quick. So here's our messenger RNA that we just drew from here. Remember, it still has our GCAP on it, our 5' GCAP. So we should put 5' there. And it still has the poly A tail that's been added to it as well at the 3' end. Now, this messenger RNA, the initial one that's transcribed from the gene, has the exon 1, intron 1, exon 2, intron 2, exon 3. These exons and introns, these are just DNA sequences, but what's the difference between them? The short answer is we want to get rid of the introns. They don't code for anything that's going to appear on a protein, so we want to get rid of them. The exons, these are the ones we want to keep. These are the ones we want to maintain on the messenger RNA because they are important. They code for the sequences that will make the protein. So what happens here, and a great deal of proteins are involved in this process to get rid of these introns. And a term your book uses, it's called, they could use a protein complex called a spliceosome. Spliceosome. Sounds like something you might find in your kitchen, but really it's something inside your cells. It comes in here and it splices out, cuts out these introns. So what you end up with is a much shorter RNA that only contains exon 1, exon 2, and exon 3. And of course we still have our 5' end with a G cap on it, and we still have our poly A tail at the 3' end. But again, it's much shorter because we just got rid of these introns. Now this can leave the nucleus. So if we were to draw this as all being in the nucleus, it can now leave the nucleus and go into the cytoplasm to be translated. Now that all by itself is a pretty cool thing that happens in our genes and during transcription. But what makes it even more interesting and shows just how creative our genes are and how we have evolved is that we have evolved a way that we can look at this sequence, or rather our cells look at this sequence and say, you know what, I could make more than one protein from this one messenger RNA. And we do that by how we cut out these introns. For instance, the way we made this one is we cut out introns 1 and 2. Right? So to make that messenger RNA, these introns were removed by the spliceosome. And we were left with exon 1, 2, and 3. And this messenger RNA makes some protein that has some function. What we know sometimes will happen now is that in some cell types, it will also remove the exon 2. It'll remove all 3 of these. When that happens, what you make is a different kind of messenger RNA. And that contains just E1 and E3. We don't have E2 anymore. It's just been cut away. We still have a 5' G cap and we still have our poly A tail. The only difference is we have removed this exon. And what this has allowed our cells to do is to produce 2 proteins from the same messenger RNA. We get more bang for the buck, so to speak. Now, why is this important? Well, it's important because as evolution moves forward, sometimes it's easier to take a pre-existing gene and reshuffle those exons a little bit to make a new protein, as opposed to creating a brand new gene. Kind of to illustrate this a little bit, we know, say like a yeast cell, single-celled fungus, has about 6,000 genes. We know humans, we'll write this down on the bottom, we have, let's say roughly 22,000 genes. This number changes frequently. When I was in school, we knew there were 100,000 genes in humans, and then more and more we studied it, we realized we were much more streamlined than we ever thought. And then let's take, say something like a fruit fly. Those pesky little things that fly around in our kitchen. They contain about 17,000 genes. When you look at these numbers, you sort of want to think that humans are more than just 4 times more complex than a single-celled yeast. You would think we would have more than 4 times as many genes. And when you think about a fruit fly, now fruit flies are kind of cool. They fly, right? We can't fly. But we still kind of think we have more complexity to what we can do than just 5,000 extra genes. What we have learned is that one of the ways that we can get more proteins made compared to a fruit fly or a yeast is we take the same genes they have, but we have different ways to splice them so that we get more messenger RNAs that can make more of a variety of proteins. So if you have one gene that can make 5 proteins, great, right? You save a lot of space, and it's evolutionarily more efficient to make proteins in that way. One thing I think I forgot to mention or I think I implied it, but I'm going to go back and make sure that it's clear, is that I drew this very simplistically with 3 exons and 2 introns. You should know that any given gene in say, in humans could have upwards of 50 to 100 or even sometimes more introns and exons in them. And we saw in this simple example here how we were able to just make 2 proteins, 2 messenger RNAs to make 2 different proteins with just these 3 exons. If you added the complexity, say, of having 100 exons, that allows you to make such a wide array of different proteins than you ever could have imagined before. All right, on these 2 slides here, we talked about how messenger RNAs are modified. The guanine cap is added to it, the poly A tail is added to it, and these introns are spliced out, and that we can have alternative forms of this splicing. It just struck me, I don't know if I used the correct term here, but when we talk about different ways we can splice something, that is called alternative splicing. For the life of me, I don't know if I actually use that term, but it's a term you should definitely know, and it's on your note-taking guide. Okay, alternate splicing. Okay, before we end this session, let's go back to this animation that we started showing with the last podcast and pick it up with RNA processing. So here's our RNA polymerase. It's making the RNA molecule, as we saw before. You're going to see here that the G cap get added to this 5' end. Here it comes, and now it's stuck on there and it protects that end from degradation. One thing they're going to do here in just a moment is they're going to start showing you what splicing is before it's fully made, and that's okay. Sometimes that occurs. So pretty soon we're going to see this messenger RNA as it's being made. The intron is going to be removed. They don't label the intron, but it's the part that gets removed. So here are the proteins that remove the intron. It comes along here. It wraps it up and cuts it and removes it. Next, we're going to see the poly A tail added. So we have our G cap at this end. Our poly A tail has occurred. They've already removed the intron. This is a mature messenger RNA ready to be translated. Exported out of the nucleus. In the cytoplasm now, it will be translated. And we will pick this up in our next objective when we talk about the molecular aspect of translation. If you have any questions over this particular objective or anything we've covered, please make sure you come see one of us as soon as you can. We'll see you in class. Bye.